So are we ready to get started?
We are. Go ahead, Brandon, and thanks for speaking today in What's New in MPMI.
Well, thank you for inviting us to do this. I'm very excited for this opportunity to discuss our article with a wide variety of people in the audience. So to put this article into context of how we were framing it when we were invited to write this paper, Tessa and I are cell biologists. For the most part, we're interested in plasmodesmata, regulation, function, and structure. And as Jean mentioned at the beginning, plasmodesmata are a fascinating system where we have learned a lot of information from viruses. And so when we were putting this article together, we wanted to try and take all of the information from the different viruses. So every virus seems to have its own set of information that have been used to garner information about the plasmodesmata. And so we wanted to compile that into a single article, a place where we could go and kind of tell a story about everything we've learned and just demonstrate how powerful viruses are as a tool for studying basic cell biology. And so just to give you a brief outline of where the talk is going to go, I'm going to start off with a brief introduction to plant viruses in general, and then dive pretty deep into the discussion of what plasmodesmata are and how we differentiate them and what they're composed of. And then we'll transition to a discussion on the different mechanisms of virus movement that viruses use to spread from cell to cell. And then finally, because I couldn't resist the opportunity, I'm going to share hopefully a little bit of new data from our work that's kind of addressing some of the standing questions in the field. And so I'm excited to not only have the opportunity to discuss this potentially and how this influences the field, but also demonstrate how the literature and all of the work that's led up to this has influenced our own research. So we'll jump right in here. Plant viruses are agriculturally very important. As the world's population continues to grow, we have to be able to feed the growing population. And unfortunately, plant disease results in estimates of around $60 billion in crop losses worldwide each year. And plant viruses are considered the second most important agent for causing plant disease. They're second only to fungi in their ability to decrease yields of plants. And so what the viruses are actually doing, it's a combinatorial effect of stunted growth. So the plants are smaller, they have decreased yield, so they produce fewer fruits. They have delayed or altered development. And finally, and by no means the least, they have altered appearances. And so if we look on the right here, we see a host of crops that are infected with a virus. And we see that they look very sick. So these tomatoes here, these eggplants, and these beans, if I were at the grocery store, these are not the pieces of fruit that I'm going to pick to purchase and take home with me to eat. So typically these never make it to market, and they end up either in a secondary market or they are disposed of. And so this contributes a lot to this annual loss in crops. And so understanding how the viruses are actually manipulating the plant to promote their spread is going to be critical in our ability to combat this massive problem that we are facing with crops. And so, like all viruses, plant viruses have to be able to spread. And unlike animal viruses, they do not do this by budding from the surface of the cell. So if we think of COVID-19, the pandemic that we're living through right now, we know that the virus is spreading through liquid droplets that are spread from us coughing, sneezing, talking. Plant viruses do not spread this way. And part of this is in part due to the fact that plant cells are encased by the cell wall. The cell wall forms a physical barrier that separates the neighboring cells from each other and provides an extra layer of support and defense for the plant cell. Now what this does is it also limits the plant's ability to communicate between cells. And so what the plant has had to do is evolve mechanisms to bypass the cell wall to exchange nutrients and information between neighboring cells. And the way that plants have done this is to evolve cell wall spanning pores called plasmodesmata. And these form direct connections between the cells and allow for the rapid exchange of nutrients and signaling molecules between the neighboring cells. Now, unfortunately, plant viruses have been able to, they're smart, and so they have evolved to hijack the plasmodesmata for their intracellular spread. And we're going to talk about what the viruses are doing to hijack the plant's natural systems, and we'll also talk about how studying this interaction has been fundamental to understanding basic plasmodesmal biology. So plant viruses use Plasmodesmata for different stages of infection. So at the early time points of infection, so this would be upon infection. Here I'm demonstrating this so we can visualize it by taking a TMV, a tobacco mosaic virus that expresses free GFP so that we can track its movement. And I've infected a benthomiana leaf with a low titer virus so that we can track the growth of these individual lesions here. And over time, after about 120 hours, we see that these lesions have actually gotten quite a bit larger, and we can see that the virus is starting to invade the vasculature system of the plants, the veins here. And this is called local movement. And this is the point where the virus is moving cell to cell through Plasmodesmata. And what it's trying to do is get to the veins of the plant so it can get into the vasculature. And once that happens, the virus is then able to quickly spread throughout the plant and move into the uninfected tissues to increase its chances of being spread to a neighboring plant. And this right here is called systemic infection. And it's a combination of long distance movement of the virus through the vasculature, particularly the phloem, and then on top of that, the cell-to-cell spread through the Plasmodesmata to invade the uninfected tissues. And so really, Plasmodesmata are critical for the plant virus to be able to spread at multiple stages of infection. So what exactly are Plasmodesmata? So on the left is a cartoon representation of what we think of when we think of Plasmodesmata. They are, as I mentioned, cell wall spanning pores. So this would be the cell wall here. And they are lined by the plasma membrane shown here in black. They form a continuous membrane between the neighboring cells. They also have this central structure derived from the endoplasmic reticulum. It's called the desmotubule. And this structure, we'll talk about briefly, is quite important for virus infection. A lot of viruses are thought to move through the Plasmodesmata in association with the desmotubule. Oh, I'm sorry, let me turn on my laser pointer. So this is the desmotubule here in gray. So this is the site where we think the viruses are moving cell to cell. And we'll talk about why that is in a little while later. But there's also this structure called the cytoplasmic sleeve, this white space here in the cartoon. And this is where we think a lot of the bulk transport of soluble molecules like sucrose takes place. So it happens through the cytoplasmic sleeve. And really, there's a lot of information that is exchanged to these pores. But there are also a bunch of proteins that are known to localize to the Plasmodesmata. And those are represented in this cartoon as these green circles and the blue circles as well. And these different proteins have different functions in regulating pore function. And many of them have actually been hijacked by the virus to promote the spread of their spread and manipulation of PD. So if we look at this cartoon, this is what we typically see in textbooks. It's the textbook example of a plasmodesma. They're single cylindrical pores. But if we look at real electron micrographs of Plasmodesmata, we see that they are more structurally diverse than what we might initially think. So we do have the single cylindrical pores, and these are called simple Plasmodesmata. But we also have instances where you have two Plasmodesmata that are really close together. So these would be considered twin Plasmodesmata. And then finally, we have elaborate structured Plasmodesmata called branched PD. And these are characterized as having multiple openings on either face of the cell wall, and the branches converge in a central structure in the middle of the wall called a central cavity. And we don't fully understand why the plant has all of these different structural classes, and we'll talk about what we do know and what we have learned from studying systems where we have specialized PD. But one thing we do know is that if you have an abundance of twinned and branched Plasmodesmata, it's indicative that these tissues are undergoing increased levels of de novo pore formation, and this is called secondary Plasmodesmata formation. So what do I mean by secondary Plasmodesmata? We can further characterize Plasmodesmata based on their origins, so where they are formed and when they're formed. And secondary Plasmodesmata are formed in the absence of a cell division event. So if we look at a cross-section of a leaf, shown here on the left, we have two separate cell files that are labeled. We have the epidermal layer and we have the mesophyll layer. And each of these cell files are clonally distinct. So the epidermal layer is from a single clonal lineage. So these cell walls here are all derived from a cell division event. And then down below, we have the mesophyll to the epidermis connection. And these cell walls did not derive from a cell division. These are clonally distinct lineages. And if we zoom in to one of these epidermal cells, the cell wall that connects the two epidermal cells is going to contain what are called primary plasmodesmata. And these are the plasmodesmata that are formed during cell division as the cell plate is being put down. The ER gets trapped in the developing cell plate, and you have pores that are formed there. You'll also have a few instances of what are called secondary plasmodesmata, where pores were added to the wall after cell division has already taken place. So that's where secondary plasmodesmata comes in. But a more clear, definitive way to determine that something is secondary is to actually look at these non-division walls between the epidermis and the mesophyll, because all of the plasmodesmata that connect these cells together are going to be secondary in nature because they arose in the absence of cell division. Now, we've already talked about how you can classify plasmodesmata based on their origin and their structure, but we can even further classify them based on their substructure. And studies with electron tomography have actually been critical for this. They identified two distinct structural classes, and they've been called type 1 and type 2 plasmodesmata. And so these are from Arabidopsis roots, typically from this paper. And what they see is that type 1 plasmodesmata are characterized as lacking a visible cytoplasmic sleeve. So they don't have a large open stream of cytoplasm visible in the tomograms. And type 2 plasmodesmata are characterized as having a large cytoplasmic sleeve. So you have the plasma membrane here, cytoplasmic sleeve, the desmotubule, and then cytoplasmic sleeve, and then the plasma membrane again in this cross-section of the EM. And this, we knew these differences were present, but what was really fascinating is that recent studies have identified type 1 plasmodesmata, the ones lacking a visible cytoplasmic sleeve, as having increased intracellular transport. So this kind of flips what we think of when we think of plasmodesmata regulation on its head, because typically we think of opening the plasmodesmata, increasing the size of the cytoplasmic sleeve, or increasing trafficking and closing down the pore for decreased trafficking. And these studies have indicated that this might not actually be the case. And so I'll talk about some more of that evidence in a little bit more detail later. So just to summarize, what I'm hoping you're seeing is that characterizing plasmodesmata is a pretty complex feature. So we can first characterize them by structure, simple, twinned, and branched. We can further characterize them based on their origin. So are they primary or secondary? And then we can characterize them by their substructures. So type 1 versus type 2, cytoplasmic sleeve or not. And so it's really hard without using electron microscopy and doing tedious analysis of this. It's hard to know when you're working with a living plant what type of plasmodesmata you're working with. And so we've had to use different techniques and different approaches to do this. And it's usually a combination of live cell imaging and electron microscopy. So we have all of these different structural classes, and that raises the obvious question of why do plants need all of these various structures of plasmodesmata for their viability? And in brief, the answer is we don't really know, but there have been some studies that have kind of tried to address this question. And one of the first ones that really sought to kind of identify distinct functions of plasmodesmata were studies that looked at the sink to source transition. So this is over the course of leaf development. These studies identified the fact that in young sink tissues, where you have a lot of import of nutrients, you have an abundance of simple plasmodesmata. So these are single cylindrical pores. And then if they looked at the transitionary stage, they saw that they had clusters of plasmodesmata, so these twinned PD, shown here. And then if they looked in the source, so the mature leaves that are doing a lot of export, they had an enrichment for branched plasmodesmata. And so coupling these observations from electron microscopy with live cell imaging, looking at trafficking capacities, they determined that they suggested that branched plasmodesmata have lower trafficking capacities or smaller size exclusion limits than primary simple plasmodesmata, so the single cylindrical pores. And so this kind of suggested for the first time that different plasmodesmata structures may have distinct functions. And this would indicate that somehow the pore structure is important for regulating these functions, and that not all plasmodesmata are homogeneous in their ability to traffic. And so later on, a few more studies were done, and one that I'm going to talk about is actually work from Tessa's postdoc, where she was looking at a chloroplast RNA helicase called ISE2. And this was one of the first times where increased intercellular trafficking was correlated with increased numbers of secondary plasmodesmata. So what Tessa found out was that when she silenced the expression of this chloroplast localized RNA helicase, she saw that there was a dramatic increase in the amount of intercellular trafficking that was taking place in her silenced plants compared to the wild type controls. And when she used electron microscopy to try and figure out what was happening with the plasmodesmata in these cells, what she found was that these silenced tissues had increased ratios of branched and twinned plasmodesmata, indicating that there was increased secondary PD formation. And this was particularly clear because the most striking increase was in the epidermis to mesophyll contact. So those walls that have only secondary PD formation happening. And so this was the first instance where we had data that suggested that you can increase trafficking simply by increasing the number of plasmodesmata in a wall, or increasing the number of branched plasmodesmata. And so we'll talk about this idea a little bit later. But if we look a little bit closer at the actual pores themselves and look at their macromolecular compositions, we see that plasma membranes are enriched in specialized lipids, particularly sphingolipids and sterols. And so these plasma membranes are actually more reminiscent to what we call lipid rafts. They're thicker membranes, they're less fluid, so there's less lateral diffusion in the membrane. And they are enriched in specialized membrane proteins as well as GPI-anchored proteins. And these have all been implicated in regulation of plasmodesmata. And in fact, recent work with a sphingolipid biosynthesis mutant indicated that if you disrupt sphingolipid biosynthesis, there's increased phloem unloading taking place in the roots of Arabidopsis seedlings. And when they started looking at these with electron microscopy, they found that this was not a function of callose metabolism. So the plasmodesmata were not being shut down or closed by callose, and it was not a factor of how many plasmodesmata there were. This was some of the evidence that led to the idea that the type 1 plasmodesmata have increased trafficking. So they found that these mutant plants had increased numbers of type 1 plasmodesmata. So those pores that lack a visible cytoplasmic sleeve. And this indicated that these pores have increased intercellular transport somehow. And it really flipped, like I said, it flipped the field on its head, because how do PD with no visible cytoplasmic sleeve have increased trafficking? Because we thought that you dilate the pore, increase transport through it, shut it down to decrease transport. And so, like I said, this is really kind of shaking. It's made it a very exciting time to be a plasmodesmal biologist, because it's kind of shaking up the story and is opening up a whole new set of questions. So now we're going to transition into that second part of the talk, where we're going to look at the different mechanisms that viruses have evolved to move cell to cell. And we're going to focus on the figure from our review. And we're going to focus this on the different instances that we've summarized here of viral manipulation of plasmodesmata. And so we'll talk about the different mechanisms that are outlined here. But one thing that is consistent about the majority of plant viruses is that they actually increase intercellular transport through plasmodesmata. And this process is called gating. So the viruses gate plasmodesmata, and it's thought that this is a mechanism for the virus to facilitate the spread of its mobile form. So if it's a ribonuclear protein complex or intact virions, it's thought that this is a mechanism to increase its ability to traffic through the plasmodesmata and spread cell to cell. And it's typically associated with specialized viral proteins called movement proteins. And just to visualize that this phenomenon, I've included some images that I took where I've infiltrated leaves from Nicotiana benthamiana with 2 agrobacterium that express 2x GFP. And in our uninfected cell, you can see that the GFP is completely sequestered in a single transformed cell. It has not migrated out from that single cell, indicated by the lack of fluorescence in the surrounding cell layers. But if we look at the cells in the tobacco mosaic virus infected plants, the GFP has been able to migrate out and has started to invade, indicating that there's increased trafficking of 2x GFP, which is a freely soluble molecule in the plant. It's now able to pass through the plasmodesmata into the surrounding layers, just kind of demonstrating that increased transport that we see with the majority of virus infections. And as I mentioned, there are many proteins that have been identified to be involved in regulating plasmodesmata. Proteomic studies have identified over 1,000 proteins that are potentially localized to the plasmodesmata and involved in regulating their function. And in our review, we talked about a handful of these, but we specifically spoke about the ones that have been implicated in virus infection, particularly in movement through the plasmodesmata. And this is an ugly slide. I'm not going to talk about all of these proteins because we would be here for a very long time. I'm going to actually focus on beta-glucanases and callose synthases. So for a long time, callose metabolism at plasmodesmata has been the end-all be-all for plasmodesmal regulation. And so we're going to focus on this because there's a lot of information out there about the callose metabolism and its role in modulating virus movement through the plasmodesmata. So it's pretty clear that callose metabolism is important for virus infection at the plasmodesmata. One of the first things that happens upon infection with virus is that you have deposition of callose at the PD. And the idea here is that the plant detects that something's wrong and it deposits callose to try and shut down the plasmodesmata. And that's based on the original school of thought of closing pores to decrease transport. But what happens at the same time is that PR genes are upregulated, specifically some beta-glucanases, so the enzymes that degrade callose and remove it from the cell wall. BG2 and BG3, which are ER associated normally, beta-glucanases are upregulated and they are recruited to the plasmodesmata. And what that indicated initially was that these beta-glucanases were upregulated somehow by the virus. So the virus was using these to increase degradation of callose and promote their spread. But later studies determined that even though they are drastically upregulated, during infection, loss of these beta-glucanases had no detrimental effect on the virus's ability to spread, and it was actually a constitutively expressed PD-associated beta-glucanase called ATBG-PPAP that was important for viral movement. And so it's unclear why these are so drastically upregulated, but it is interesting that we still see that if you lose specific beta-glucanases, you do see increased accumulation of callose at plasmodesmata, and this increased callose deposition does seem to delay specific virus movement, best exemplified by work with tobacco mosaic virus and potato virus X. So the degradation of callose does seem to be important, but it's not necessarily the beta-glucanases that are specifically upregulated during virus infection. And then another study further demonstrated this by overexpressing a type 1 beta-glucanase from tobacco by incorporating the coding sequence for this protein in the virus itself and then infecting the plants. They showed that the presence of this beta-glucanase increased the rate of systemic spread of the virus itself and helped promote its rapid spread throughout the plant, further supporting the idea that callose metabolism is critical for, or is important for promoting virus movement. And so, as I said, we don't really know what exactly is going on, but it does seem to hint that callose metabolism is one of the critical steps in this. And so most viruses encode specialized proteins for cell-to-cell movement called movement proteins. And these proteins are thought to be some of the factors that are involved in triggering this process of increasing the transport through plasmodesmata through gating. And movement proteins are very diverse. They come in different shapes, different sizes, all sorts of conformations, but they all share some similar features. One of the primary things is that one of their main goals is to target the viral genome in some way or another to and through the plasmodesmata. And if we look at this confocal image here on the right, this is tobacco mosaic virus's MP30 labeled with GFP, and we see these characteristic PD foci labeled in the cell wall here. And this is pretty typical for movement proteins. They accumulate in the plasmodesmata and have been really important in our understanding of basic features of pore function. But these movement proteins are also typically able to bind specifically, or not specifically, but bind to RNA to help promote spread of the genome between neighboring cells. And they also typically interact with membranes. And so this is best exemplified with TMV, where the movement protein interacts with the ER and is targeted to the plasmodesmata and helps, we think, facilitate movement of the viral genome through the plasmodesmata through its association with the desmotubule. And so that helps promote spread of the virus in that way. And then some viruses encode multiple movement proteins, and these are really fascinating. So these are best exemplified by the triple gene block viruses, and they have 3 movement proteins, and each of the proteins plays a distinct role in virus movement. And so we'll talk about some of those details briefly in a few slides. So how exactly do viruses gate plasmodesmata? So how are they increasing trafficking? And so in our review, we talked about 3 specific possibilities. The first one was through the most common idea is callose degradation to dilate the plasmodesmata. But the other ones we talked about were altered macromolecular composition to allow changes in trafficking. So this would implicate the plasma membrane. So if you could change the plasma membrane composition, lipids, biosynthesis, and stuff like that, you could potentially change the pore function, or you could sequester proteins away from the plasmodesmata or recruit new proteins to the PD. So altering the plasmodesmata composition there could potentially be a way to gate the plasmodesmata. And then finally, we talk about drastic remodeling of pore structure. And so this is best exemplified by specific viruses that induce tubule formation, and we'll talk about that mechanism, as well as viruses that dramatically remodel the ER at plasmodesmata. And those were 2 of the things that we've highlighted in our figure, and we'll break those down in a few moments. But basically, some of the major changes that we think about would be the removal of the desmotubule. So just get rid of the desmotubule to increase flux through the pore potentially. And so if we move into the first mechanism where we have degradation of callose, if we look at the cartoon from the very first couple slides, these black lines here in the cell wall represent callose. It's a beta-1,3 glucan that can be deposited at plasmodesmata and then removed. And it's the degradation by beta-glucanases that is typically thought of as a mechanism to increase transport by dilating the plasmodesmata. And it's thought that this is through the activity of the specialized movement proteins. So the movement protein is somehow able to trigger degradation of callose, dilate the plasmodesmata, and increase transport through the PD. And with the new insights from the tomography, this is now in contention. So is it really that they're dilating the plasmodesmata? But if we really look at electron micrographs from plants that are expressing the viral movement protein, so these are actually from transgenic plants expressing MP30, so TMV's movement protein, we can see that the plasmodesmata themselves are indistinguishable from the plants, the wild type plants that are not expressing MP. They don't appear to be dilated. They don't have major changes to their structure. So this was kind of already there to kind of suggest that maybe the dilation of the plasmodesmata is not as important as we initially thought. And if we look at potato leaf roll virus, which is actually a phloem restricted virus, if we look at meaning that its entire life cycle takes place in the vasculature of the plant. If we look at plasmodesmata in the mesophyll cells, they have typical diameters. They don't appear to be dilated. They don't appear to have any altered conformations. And the only instance that I've ever really seen plasmodesmata that look to be maybe a little bit larger than the normal with electron microscopy in the literature would be when we look at the plasmodesmata in the vascular tissue in potato leaf roll virus MP17 transgenic plants. And here's one example of that, where you have these darkly stained structures here that appear to have an accumulation of something in the plasmodesmata that the heavy metals are interacting with to give rise to this dark staining. But this is the closest example of a dilated plasmodesma I've ever seen. But what you can see is that there's not a visible cytoplasmic annulus here. I can't differentiate the desmotubule from the ER here, from the cytoplasm here. So really that dilation of the pore is still kind of a contentious question. one thing that we have learned, and it was kind of hinted at in that last slide, is that specific movement proteins interact with specific plasmodesmata. And so some movement proteins are able to interact with a variety of plasmodesmata, and then other movement proteins have specialized targeting for specific classes. And this is further indicative that not all plasmodesmata are created the same equally. They're not the same. There's not a homogeneous population, and they may have distinct functions. And so to kind of demonstrate this, if we look at Arabidopsis seedlings that are expressing either TMV MP30 GFP or potato leaf roll virus MP17 GFP in the roots, we see that MP30 is actually able to label the plasmodesmata in the roots. And this is particularly interesting because Arabidopsis roots lack branched plasmodesmata. And early studies indicated that MP30 was only able to label accumulated specifically in branched plasmodesmata. And here in the roots, we don't have an abundance of branched PD, yet MP30 is able to localize to the plasmodesmata and form those distinct foci that we see in PD. But if we express the phloem restricted movement virus PLRV as movement protein MP17 in the roots, we fail to mark the plasmodesmata, and you get a diffuse signal throughout the cytoplasm. And if we were looking at the zoomed out image from this same figure, you see these large aggregates of the fluorescent protein that are just accumulating in the cytoplasm. And this indicates that MP17 is only able to label specifically the branched plasmodesmata. And the same phenomenon can be observed if you express MP17 in sink leaves and compare those to the source leaves. In the source leaves, where you have undergone the sink to source transition and you have branched plasmodesmata, you will have labeling of the PD in the cell periphery. In the sink tissues, you have the aggregation of MP17 and failure to label plasmodesmata. So this strongly suggests that specific movement proteins have specific functions and interact specifically with distinct plasmodesmata. So now we're going to move into some more specific examples of how viruses move cell to cell. And the first one we're going to talk about is kind of an oddball in terms of plant viruses. It is turnip mosaic virus. It's a potivirus, and it is thought to move cell to cell as vesicular replication complexes. So this is completely distinct from the majority of viruses that either move as ribonucleoprotein complexes, so a few proteins in their genome, or as fully formed virions. And we'll talk about that last one in a few moments. These guys are actually thought to move, or turnip mosaic virus is thought to move as a mobile vesicle that is derived from the ER and contains specialized viral proteins, the 6K2 protein and the TUMB replicase. And these associate with the genome and traffic the vesicles to the plasmodesmata and potentially and presumably through the pores and into the neighboring cells to spread. And if we look at confocal images of this, and here they've labeled 6K2, that viral protein with GFP, and they've stained the plasma membrane, they observe that some of these, indicated by these white arrows, some of these vesicular bodies are associated quite closely, and they suggest tethered to the plasma membrane. And when they looked at this interaction a little bit closer, and they expressed 6K2, this time fused with M. cherry, alongside a plasmodesmal marker, PDLP1 GFP, they saw that these red, or the 6K2 vesicular bodies, were tightly associated to the plasmodesmata, suggesting that these vesicular structures are targeted to the plasmodesmata, anchored there, and are somehow implicated in the spread of the virus but through the PD. And so this is a really fascinating model for virus movement because it's completely different than anything else that's out there in the literature right now. And so there's a lot to be learned from this mechanism, and it just kind of illustrates how viruses will do whatever it takes to move, and they have evolved multiple different mechanisms for this. So now if we move on to the next panel from our figure, here we're looking at a potato virus X, and this is an example of the TGB viruses, the ones that express multiple movement proteins. And this is one of the first examples of dramatic changes to core plasmodesmal structure that we see and have verified evidence from electron microscopy of this. The TGB proteins remodel the cortical ER at the plasmodesmata and form these ER-derived caps that presumably are involved in loading of the viral replication complexes to the plasmodesmata and helping promote spread. And our cartoon was inspired by electron micrographs such as this one, where we see that remodeled cortical ER in close association with the plasmodesmata here. So here we're looking at a cross section of the pore, and we can see this through the wall. So we see the little spots that correspond to plasmodesmata. And so this really is one of the best examples of major changes that take place at the plasmodesmata in their core structure that might be involved in regulating function. And then we'll move briefly into the tubule forming viruses. So these are also really cool because they are another example where viruses actually dramatically alter the structure of plasmodesmata to help promote their spread. And in this system, the movement protein, we're looking at grapevine family virus in our figure here. The movement protein is made in the ER and is matured through the Golgi and exocytosed out to the plasma membrane, where it accumulates and then will polymerize into these proteinaceous tubules that actually pass through the plasmodesmata. And these tubules allow for the movement of fully formed intact virions through the plasmodesmata. And this is completely another example of something that's different than what we were talking about with TMV, where you have a mobile RNP, ribonuclear protein complex. Here the virus is fully formed, and in order to do that, you have to be able to change the plasmodesmata structure because the virus is too large to fit through the pore with the desmotubule. And one of the features that we think happens at the plasmodesmata when you insert the tubule is that you actually remove the desmotubule from the plasmodesma. So you open up the pore by inserting basically a straw through the pore where the viruses can move through. And if you express the movement proteins of these viruses in protoplasts, you see that you have these protrusions that stick out from the protoplasts, plasma membrane. They spontaneously form in the absence of plasmodesmata and stuff like that. And if we purify some of these and we look at them, what we see is that they're actually tubules. These are the tubules that pass through the plasmodesmata, and they contain intact virions. So these are pure, in these electron micrographs, these are purified tubules containing the virus, and we can see that here. There are also electron micrographs out there that show these tubules in association with the plasmodesmata. And so they really, this is a fascinating way that the viruses do in fact alter the structure of the pore by disrupting core components of the PD. All right, so for the last few minutes here, I want to briefly introduce you to a few parts of my own research that are trying to answer some of the questions and provide an alternative explanation in light of what we know about the new structural classes of plasmodesmata and how viruses may not be gating plasmodesmata by dilating the pores. All right, and so I've already told you that Tessa's postdoctoral work indicated that you can increase intracellular trafficking through plasmodesmata by simply increasing the number of secondary or branch plasmodesmata. You can increase transport that way. And in fact, in our lab, we've silenced a bunch of different genes. The identities of those are not particularly important for today's talk. But when we silence these genes and we look at their PD density, so how many plasmodesmata there are per unit of cell wall, and compare that to how much intracellular trafficking we have in these tissues, we see that there's a pretty striking correlation between the number of plasmodesmata and the amount of intracellular trafficking. And so this got me thinking, is it possible that gating could simply be a result of increased numbers of plasmodesmata in our infected tissues? And so to try and look at this, I developed an assay that I'm able to use to fairly quickly look at plasmodesmata distributions in our tissues. And so here I transiently express a plasmodesmal marker. For the first set of experiments, I'm using PDLP1, which is a plant protein that will localize to all of the plasmodesmata. And I'm able to go in and measure the cell wall length, and I know how thick they are, so I can get an area of cell wall, and then we can quantify the number of foci that we see. So these foci correspond to pit fields of plasmodesmata. So this allows us to quickly assess the PD density of our tissues. And when I look at, here I'm showing TMV infected leaves from the primary infected leaves. So 3 days after infection, we see that there's a 14% increase in the number of pit fields in TMV infected leaves. It's a moderate increase. And actually, when I look at systemically infected leaves at around 10 days, I actually see a more striking increase where we're having closer to 25% increases in the number of Plasmodesmata, suggesting that, yeah, we actually do have increases in PD density when we infect with the virus. And then if I use another movement protein, MP17, so that's that viral movement protein that we showed that was shown to specifically label branched Plasmodesmata. If we look at that in our infected leaves, we actually see a 25% increase in the amount of the proportion of Plasmodesmata that are branched, indicating that we have elevated levels of secondary Plasmodesmata formation happening during virus infection. So this suggests that somehow the virus is able to trigger I would assume the plant's endogenous mechanisms for forming Plasmodesmata. It's able to trigger that and increase the number of PD, potentially serving as a mechanism to increase trafficking. And so something that's really exciting that we've seen is that if we take the viral movement protein from TMV, so the agent that is believed to be the source of gating, so the protein that gates the Plasmodesmata, if we express that in an uninfected leaf, we see that same 14% increase in the number of Plasmodesmata that we see during virus infection. And so it's striking that we were able to phenocopy basically the effect of the virus infection on PD density simply by expressing the movement protein. So we think that this is some pretty solid evidence to suggest that gating could be simply a result of increasing the number of Plasmodesmata in our infected tissues. And in fact, we've actually tested this on a variety of viruses. We've done tobacco mosaic virus, tobacco rattle virus, and we've currently done 2 Jimny viruses, and we're working on doing a Potex virus and a potivirus in the near future. And we're seeing these same increases across the board. So it seems to be a conserved mechanism that viruses are able to increase transport through the Plasmodesmata by simply increasing the number and triggering secondary Plasmodesmata formation. So just to summarize my work, what I'm saying is that virus infection does seem to increase Plasmodesmata formation in Benthamiana leaves. And this increased formation is specifically in branch Plasmodesmata, and that suggests that it's secondary pore formation. And expression of TMV MP30 alone, in the absence of the rest of the virus, is able to induce PD formation. And so this actually, we think, is a potential new mechanism or a new model for how viruses gate Plasmodesmata. And so to put all of it into context, what I hope you've seen from our discussion of the review today is that viruses have evolved to utilize different mechanisms for manipulating the Plasmodesmata. They've also revealed many of the details of Plasmodesmata structure and how that's important for regulating their function. And I hope we've been able to illustrate simply how important studies with viruses have been on understanding basic principles of plasmodesmal biology. So something I failed to mention earlier on is that even some of the fundamental studies looking at macromolecular transport through Plasmodesmata were first done with viral proteins and studying viruses. So foundational work was done in these virus-affected plants. And so it's been a very, very powerful tool for studying PD. And I think it's likely to continue to be that way, where we have that system where we can continue to study and learn more about different features of Plasmodesmata simply by studying the virus-Plasmodesmata interactions. And so I'd like to quickly thank my wonderful boss, Dr. Tessa Birch-Smith, our current and former lab members, as well as MPMI for allowing me to give this talk on our review and allowing us to write the review, because it was a wonderful experience that helped benefit me in the long run. So it was really helpful, and I am excited to now jump into the discussion and answer some questions.
Thank you, Brandon. That was a really terrific talk, very interesting. I've always been fascinated by this whole idea of Plasmodesmata, how it works. Very clear. So if folks want to ask questions of Brandon, please type them into the question and answer, and we will start to answer them. Before we do, I just want to remind you that this review was part of a special focus issue that we did on the cell biology of plant virus and virus vector interactions that was guest edited by Tessa Birch-Smith, who's my co-host today, as well as Aiming Wang and Yi Li, all editorial board members on the MPMI editorial board. So, okay, so let's let me start by reading the first question here, and then Tessa and I will take turns. So Apple Chew writes, are the different types of Plasmodesmata consistent throughout all plant species? Brandon.
So that's an excellent question. So we do see predominantly that different plant species do have different types of Plasmodesmata. In terms of the type 1 and type 2, I think they've only been currently observed, I may be mistaken, but I think it's only been currently observed in Arabidopsis. That's simply because this is a very new proposal, and we've just now started to apply these advanced electron microscopy imaging techniques to look at the Plasmodesmata. So I wouldn't be surprised to see that these substructures do exist in other plants. We just haven't really looked at them in that great of detail yet. But by and large, the majority of plants do have simple, twinned, and branched Plasmodesmata.
Tessa, you need to unmute.
Yes, that would be helpful. Okay. So the second question comes from, pardon me for butchering your name, Guillaume Silva Martins, and he asked, what do we know about changes in Plasmodesmata during viroid infection?
That is a very fascinating question. Honestly, I don't think we know a lot about them. We know that these infectious RNA molecules do move through the Plasmodesmata, but they don't encode any proteins. And so really there's not a lot that we do know about them. And it's, I think, a fascinating area of future experiments and research that there's a lot to be learned from those viroids, because what are they doing? I mean, they don't make any proteins. So how are they, if they are, how are they changing the Plasmodesmata? That's a fascinating question.
Excellent. So Sridhar Ranganathan asks, how and where the possible incompatibility between the virus and host cell, the Plasmodesmata region, is expressed. So yes, wondering about incompatibility between virus and host, I'm guessing in perhaps in a resistant line. I think that's what this is going at.
Right. So many of these situations, they have the host encodes other defense mechanisms for the virus that would be implicated in this. In terms of the Plasmodesmata, I'm not sure, it's not particularly clear where that would be, but it would potentially be that one potential mechanism would be that the virus fails to gate the Plasmodesmata. But really it comes down to the receptors and the different defense mechanisms that plants have evolved to combat this. And Tess is honestly the expert in the defense between the receptors and viruses. So if she has anything else to add to that, I think she would be very beneficial for that question in particular.
I don't know if we have any special mechanisms operating at the Plasmodesmata during incompatible interactions, during defense responses. It seems like everything happens before we get to the Plasmodesmata. You stop it from getting to the Plasmodesmata and not through the Plasmodesmata.
Yes.
Go
ahead.
There's a request for Brandon to stop sharing his screen. I'm not sure what that's about.
Yeah. We can stop sharing for now, and then if you need to go back to it to answer a question, we can always open that back up again. Yes. So Kwanju Lu says, one of a key feature which has been demonstrated before is the ability of virus movement proteins to help transport larger proteins like 3xGFP. And that's why people believe movement proteins can dilate the Plasmodesmata. So how do you think this correlates with the increase of secondary Plasmodesmata?
So that's an excellent point and an excellent question. So the idea here is that while the dilation of the Plasmodesmata was initially thought to allow the passage of larger molecules through the Plasmodesmata, I think what could be happening is that by increasing the number of pores, you decrease the amount of time that it takes for the larger proteins to move. So they have more options to move through the plasmodesmata, and so it increases over the specific time that you're looking, because you have more options for GFP to move through. The larger molecules take less time to move through the plasmodesmata because there's more options. Because if we look at, like I said, those dilated, the tomogram pictures where we have the no visible cytoplasmic sleeve, I mean, it's a fascinating question, and it kind of breaks my brain when I think about the idea that these pores without dilated cytoplasmic sleeves do seem to have the increased trafficking. So briefly, I don't know the exact answer, but I think it has something to do probably with the time, because if you look at the movement of larger molecules, so for 2x GFP, if you were to look at a later time point than you do for some of these studies, the 2x GFP would eventually move into those neighboring cells. It just moves slower. So I think it could potentially be that you have more opportunities for the protein to move, and so that they move further more quickly, and it's not necessarily the size exclusion limit of the plasmodesmata themselves. It's just with the PD that are there at the time, the larger molecules have less options to move, and so they move a little bit slower.
Okay, our next question comes from Nico Cecchini, pardon me. He asked about PD specificity. Is it possible to increase movement of some molecules but not others?
Oh, that is another, all these questions are really good. So it's unclear. I think potentially if what we were proposing is true, that different plasmodesmata have different functions, then the plant could very well have mechanisms in place to regulate specific transport. So if there are certain proteins that only move through branch plasmodesmata, if you increase the number of branch plasmodesmata, those host proteins could potentially have increased transport. And the same could be said for the other direction. But for things that move by diffusion, potentially maybe not as likely. So like sucrose, it might be a little bit harder to specifically upregulate sucrose transport through the plasmodesmata without upregulating transport of other diffusive molecules. So that would be, it's a fascinating and interesting topic to think about, but it's probably comes down to the specialized functions of PD.
Yeah, I just want to add here that one of the challenges with studying the plasmodesmata is that all our assays on trafficking capacity and permeability tend to be bulk assays. It's hard to analyze individual cells or individual molecules or cargo just because of the nature of the system that you have to work with intact tissue. So the jury's still out on that question. But yeah, Brandon is right. It's possible that you can regulate larger molecules, but you would have a harder time regulating smaller molecules. For example, sucrose, which is so small, it should be able to diffuse regardless of, unless you have a very tight closure happening. Yes. Okay. I have another question now from Ziv Spiegelman. He says that movement proteins were shown to associate with microtubules. How would microtubules be incorporated into your model for this new idea you have for plasmodesmata formation?
So that's a fascinating question. So like I said, all these questions have been great so far. And the movement proteins of different viruses have been, like you said, have been shown to incorporate or interact with microtubules. They also have been shown to interact with actin and other cytoskeletal elements. And what I think might be happening with the microtubules, it's hard to say. So we know that the microtubules in plants are in close association with the periphery of the cell. So it could be that there is specialized targeting that's taking place on the microtubules for cell wall modification and plasmodesmata remodeling and pore formation, but there's no evidence out there really right now to suggest that this is the case. So I don't have a solid answer for that question, but I think it's definitely something that is worth investigating. So I do see in some of I've been doing some tomography, and I do see that in my tomograms where I have plasmodesmata, I typically have a lot of microtubules in close proximity to them. I don't know what that means in terms of this phenomenon that you're referring to, where they're interacting with the microtubules, but they do seem to be enriched at the plasmodesmata. I don't know specifically what they're doing, but it's an interesting idea.
Interesting.
Yes.
Yeah.
So one
As
idea
I
we
hear
have is that we know that microtubules are involved in cell wall remodeling, targeting the cellulose synthase complexes to the cell wall. So it's possible that if we're going to be in a situation where you're adding plasmodesmata to a cell wall, you will need to somehow modify the cell wall. So the microtubules may come into play in a mechanism where you want to modify the processes by which you're shaping the cell wall. So that's several studies away, but that's one possibility we're considering.
Interesting. One of the things I'm struck by as I listen to this is the big difference between studying plasmodesmata and things like channels, where you can just strip away the cell wall, you can get rid of neighboring cells, and you can study it just fine. You can study it in yeast. And with plasmodesmata, it's all about the structure of the plant. It's so integral. You need intact tissue layers, you need the cell wall, right? Without the cell wall, can't really have a plasmodesmata.
Exactly.
So yeah, very, very interesting. Okay, well, I'll move on to Wenjia Hao's a question. What are the ratios between primary and secondary plasmodesmata in healthy and virus-infected plants?
So that's a really tough question to answer, because so differentiating between primary and secondary plasmodesmata, without knowing exactly the cell wall and the cell types you're looking at, it's really difficult, because if you look at primary and secondary plasmodesmata, they're structurally, you can't for sure say that this is primary and this one is secondary, because as I said, you can have the cell wall be a result of division, and then you can then add plasmodesmata. And our studies where we're looking at the PD distributions in virus-infected plants, we haven't actually looked at the epidermis to mesophyll contact, so that would allow us to specifically look at that. That's in the works right now in our next series of experiments with EM. In order to do that, we'd have to use electron microscopy. And so we'll be doing that in the very near future, but we do see that there are elevated levels of branched plasmodesmata. So that would indicate that there are increased numbers, but our assays that we're using so far only look at the distribution of what are called pit fields or clusters of plasmodesmata. So we can see that there are increased numbers of pit fields with enrichments in branch PD in virus-infected plants. But that doesn't really tell us anything about the ratios of the actual branch PD to or secondary plasmodesmata specifically to the primary PD. So that's something that we'll definitely have to look at in the future.
Thank you. So Shihao Zhang has a question. He wonders whether the movement protein promotes the PD localization of the PD marker that you've used in your experiments.
So for the movement protein modulates the recruitment of PDLP1. Is that that question?
Yes, yes.
To my knowledge, no, they do not seem to be targeted to the plasmodesmata through the same mechanism. There are movement proteins that do utilize a similar pathway, but it's not the MP30, the TMV MP30. Some of the tubule-forming virus movement proteins, I want to say, utilize a similar pathway to get to the plasmodesmata as PDLPs, but the TMV's movement protein does not seem to interfere with the localization of our marker.
Okay, so Juan Carlos Diaz-Ricci asks, do you have any insight about whether the viral infection increase is due to the change of PD structure or also to the induction of new secondary pore formation?
So that's interesting. I think that's going to be a hard question to answer, because as Tessa mentioned, we have to, in order to study these details, to study the plasmodesmata themselves, you have to look at fixed tissues and compare that to, for electron microscopy, and compare that to functional assays where we're looking at bulk transport. So it really makes pinpointing the causative agent of the increases in spread very difficult, because you're looking at the total, all the plasmodesmata in a wall during a live cell imaging event, and then when you go to the electron microscope, you're looking at one specific time point frozen in time, and you can only gain information about that one instance. And so it makes it really difficult to tease apart those details. But that's a tough one.
Okay. So a question from Yongling Jiang, and he asks, he says that the increase in plasmodesmata should require the synthesis of additional membranes. Have you tried some lipid synthesis inhibitor of silencing of ER-associated proteins, which may lead to the suppression of your PD increase?
Right. So that is I like this question a lot. So that's something I've been thinking of in my head. I think it's something that I really like to do, given the time. But I think it's definitely something we need to look at, because you're right. The incorporation of these new plasmodesmata is going to require the production of new membranes. And I think it'll be interesting to look at, not only, this is another area where I think the viruses will be really important, because we have now a system where we have increased levels of formation. So now we can start trying to identify mechanisms by which the plasmodesmata, the cell says, you're going to make a plasmodesma here. So you're going to use this mechanism to insert the ER into the newly formed pore. So there's a lot of questions that I think can be answered with these studies in the future. And that's an excellent part of that. And I think it'll definitely be somewhere that we will look at in the near future.
Okay, Andrea Paterlini asks, since you observe an increase in PD numbers in response to viral infection, do you also detect feedbacks in cellular metabolism, AKA to produce those membranes? This is related to the previous question, to produce the membranes or modify the wall in order to achieve twinning or de novo drilling?
Right, so that's a good question. We haven't actually looked. We haven't gone in to look at the actual cell metabolism going on involving this. But we are currently in the process of trying to identify the processes that would be involved in doing this, so that can provide us with some extra insights into how this is happening and what needs to take place in order to form the new plasmodesmata. And so we're starting to start to dive into that direction, where we're looking at the metabolism that takes upstream of plasmodesmata formation. So we're in the process of doing that, but we haven't done it yet.
Okay, so Amit Joshi asks, well, first he says, great talk. Then he asks a series of questions. I think he's trying to get away with it. So he asks, do all PD have desmotubules? Does loss of desmotubules affect PD function? And do you know if ER shape regulates PD function? So it's all about the ER here. Yes,
so those are some really interesting questions. And so I'll first start by saying thank you for the compliment. The first question about, do all plasmodesmata have desmotubules? That is we don't know. In our lab, we use high pressure freezing to fix our tissues. It's the gold standard in fixation of plant tissues for electron microscopy. But unfortunately, one of the artifacts, it has fewer artifacts in general compared to chemical fixation, but one of the few artifacts that it does have is that it has very poor staining of the ER membrane. And so in our electron micrographs, we rarely see the desmotubule, and we rarely see the ER stained by these heavy metals. And this just is a function of the chemistry of the membrane itself. So I don't know that there's any direct evidence to suggest that all plasmodesmata do have PD, but typically you do see them. You do see desmotubules in the majority of plasmodesmata. The second question was, what happens to pore function when the desmotubule is removed? And so that is best exemplified by some of the early studies with the tubule-forming viruses that actually indicated that insertion of the tubule, and so that disruption of the plasmodesmata, actually had decreased trafficking of fluorescent dyes and stuff. And so that suggests that potentially if you insert a tubule and remove the desmotubule of the plasmodesmata, that actually hurts the plasmodesmata itself. It's really only good for moving the virus from one cell to another. But I can't think of another example where you have, other than the tubule-forming viruses that we see decreased trafficking, where you have removed the desmotubule and looked at the effects of virus infection. And then finally, the last question was, how does the shape of the ER relate to plasmodesmata function? And that's I don't know. I think that's something that is going to again, be coming in near future, where we start looking at those mechanisms for how the cell says you're going to make a plasmodesma here. Because I think targeting of the ER to specific sites is going to be critical for formation of the plasmodesmata. And there's evidence to suggest that membrane contact sites are critical between the ER and the plasma membrane are critical for plasmodesmata function and regulation. So I think it'll be interesting to look at this in regards to how plasmodesmata are made as well. But
great answers, Brandon. One of the things that really strikes me as you're discussing these is how much is still not known. We've been studying TMV since I don't know when, but a long time, and how much we've learned and how much is completely unknown about plasmodesmata and the role of these desmotubules and what viruses are doing, and the fact that viruses might all be doing something completely different. And
yes.
I hope the people who are listening are realizing how many different areas and avenues of future research are open, because there's still so many really fundamental questions here. So moving on to the next question, Ning Yu says, movement proteins of many viruses can increase the number of plasmodesmata. So is there a conserved host protein that interacts with these MPs?
So that's an excellent question, and it's another we don't know. I think this is where we will have more insights, like I said on the conclusion slides. These viruses are going to be powerful tools to continue to learn about plasmodesmatas, because if we can start identifying those host proteins that might be involved in this process, so the recruitment and formation of plasmodesmata, and if we can identify proteins that are used by multiple viruses to do this, that'll provide some of the groundwork that's needed to start to address that question. Because currently we don't know how secondary plasmodesmata are added to the wall. We have no idea what proteins are involved in that process. How do you drill a hole through the cell wall without rupturing the cell integrity? We don't know how that happens. And so until we figure out the players involved, I think that's going to be a difficult question to answer. And so we're starting to work on that now. And I think viruses will really prove to be useful for this.
Okay, Brandon, our next question comes from Aurelie Rako Tondrafara. And she says that you've mentioned different types of plasmodesmata in the sink versus the source tissue. Do you think such differences in the types of plasmodesmata would affect the ability of a virus to infect various tissue, or to limit their movement to only particular tissues?
Yes, so I think that's an excellent question. I think some of the best evidence for this comes from viruses that are actually sequestered in the vasculature. And so the idea here is that the movement proteins are only able to interact with specialized plasmodesmata. Potato leaf roll virus's movement protein is only able to interact with the branch plasmodesmata, as we talked about in the talk. So I think the fact that the virus is sequestered in the vasculature may indicate that it has some specialized ability to only interact with specific types of plasmodesmata and is unable to move out of the vasculature into the other tissues. So that kind of hints at that question. But I think it's to be determined whether or not other viruses that are not restricted to the vasculature are able to manipulate the plasmodesmata in some way to invade the sink/source tissues at different degrees. So I think that's definitely an interesting question, and the potato leaf roll virus is a pretty good example of where we do have specialized functions of their movement protein to interact with specific PD, and yet it's only sequestered and they only seem to have functions there. So I think that's kind of hinting at that.
So our next question is by Shifeng Zhu. Are there any differences between different plant species? For example, size exclusion limitation?
So I think yes, there are some indications that there might be slight differences, but by and large, I don't know, because different plants, I think the size exclusion limit of plants is probably going to be pretty fluctuate quite a bit across developmental time frames. So as the plant gets older, the size exclusion limit is going to change. Under different stress conditions, it's going to change. Under simply different light conditions, it'll change. So I think there are a lot of factors that are going to confound that issue and make it very difficult to really get a definitive answer for that. But by and large, I think Plasmodesmata typically have a fairly narrow range in their size exclusion limits. And I think it's pretty consistent across different plant species. Some plant species have more Plasmodesmata than others. And that's something that could be a completely different talk. But so that could influence the amount of trafficking that takes place in those plants versus those that have fewer Plasmodesmata. But there's a lot of factors that go into play there.
So there's a question. I guess this is sort of a philosophical question. So is the mechanism of new Plasmodesmata formation clearer now?
No. That would be like my dream. So part of my thesis is trying to figure out how you do this. Now I think we have a good system where we have increased amounts of Plasmodesmata formation that we can easily now go in and try and identify the players involved in this. So if we disrupt, I'm sorry?
Don't say easily.
Oh yeah, more, I won't say easily. It's not gonna be easy, but I think it'll be, we have an opportunity to do it now that we did not have before we identified a system where we have, where we can trigger elevated levels pretty easily, just simply by infecting or expressing with the movement proteins. So we can trigger this, and then we can look at players that might be involved in this process. So clearer now, no, but hopefully in the near future, yes.
Yeah, it's a very nice tool you have now to be able to increase that. That's going to be interesting. So next question, Stephanie Flieger asks, for viruses inducing tubule formation, do we know the mechanisms that underlie desmotubule disruption?
I don't think that we know definitively how it happens. We do know that it's going to require some type of mechanical energy to like break to make the ER discontinuous between the neighboring cells. But really the details behind how that happens is not clear. It's also possible, we saw in the protoplastic expression plants, and there's some evidence to suggest this, that tubule formation actually forms, it happens spontaneously in the absence of Plasmodesmata. So if you don't have any Plasmodesmata, the tubules will still form and they will stick out at the surface. So it's possible that it could just simply be the movement of the tubule, the growth of tubule and proliferation that just pushes it out of the way. I don't know, but I think that's, as all of these questions have been, is an excellent question and further highlights the black box that is Plasmodesmata.
Okay, what happens in double infections when you have branching or tubule forming viruses? Will they increase the chance of spread of movement deficient viruses?
So there is some evidence to suggest that if you take a virus that is unable to move, it does not encode a viable movement protein or something like that, and you co-infect that with a virus that is able to move and is able to gate Plasmodesmata, that the non-mobile form of the virus does have increased movement from the infected cells and increased spread. But in terms of the Plasmodesmata feature here, I don't know. I think, yes, the ability of one virus to increase the movement of a non-mobile virus has been documented in the past. There is evidence to suggest that, like say, a phloem-restricted virus, if you infect with TMV, there's increased movement of the phloem-restricted virus out of the vasculature into specific cell types, suggesting that the TMV is able to somehow manipulate the PD to help facilitate movement of the restricted virus out. Whether that's a function of the ability of TMV's movement protein to bind non-specifically to the viral genomes and then traffic it out, that's not known. Because MP30, the viral movement protein of TMV, it binds RNA non-specifically. So it could be simply the movement protein is able to shuttle the other virus out as well. But I don't know if I don't think that's been demonstrated specifically.
So Harshvardhan Gaikwad asks, please explain how movement of bipartite Gemini virus occurs through Plasmodesmata.
So Gemini viruses are weird. So they're the DNA viruses, and We didn't talk about Jimmy viruses very much in our review. We kind of focused on the RNA viruses.
Even among RNA viruses, there's such diversity of strategy.
Yes, so that is a question that I'm not sure I have an answer for. Tessa, if you have any insights into that, I don't.
I don't know. I mean, there has been some work done, but I confess that I can't recall right now. So that's something we might have to go and look up.
Yes,
Again.
definitely, because we are moving into using Jimmy viruses in some of our experiments. And from my data with those viruses, they seem to do the same thing with the Plasmodesmata. There seems to be an increase in the Jimmy virus infected plants. We're looking at African cassava mosaic virus, and it's actually the increase that I've seen in the systemic leaves is, I don't want to say more, but it's comparable, if not a little bit more substantial than we see in TMV. So I don't know exactly what that means per se, but they do seem to do the same thing in terms of the Plasmodesmata formation that we're seeing.
Okay, Francine Govers asks, is it known if fungal or oomycete pathogens can modify the structure of Plasmodesmata for their own benefit? For example, transfer of effectors to neighboring cells?
So that is a fascinating question, because there are specific receptors on the Plasmodesmata themselves that can be triggered and correspond to signaling from the Plasmodesmata to the nucleus during fungal infection. But in terms of the fungus, and these will typically shut down Plasmodesmata by inducing callose deposition of Plasmodesmata. But in terms of the fungus changing the Plasmodesmata structure, I don't know if there's a lot of evidence for this, but they definitely do seem to have an effect on the Plasmodesmata themselves. And there are some instances where effectors definitely do move to and interact with the Plasmodesmata and modify their function. So that's an excellent question, and is definitely room for exploration there.
Okay. Gulzamal, pardon me, Mukianova asks, is there any evidence of Plasmodesmata gating or other changes during the HR response?
Yes. So I think the general idea is that during HR, you have shut down the Plasmodesmata to just kind of sequester, I don't want to say sequester death, but like sequester it so that it dies in the region that's infected, and everything will just stop importing, the transport out stops, and the idea is that you just shut it down. And so the transport out of Plasmodesmata is generally decreased, or out of these regions are typically decreased during the HR response.
Okay, Luke Sofer asks, I thought Plasmodesmata were coming from cell division, but your last results seem to suggest another way of PD formation. Am I right to think that?
Yes, so I mentioned it briefly early on in some of the early slides that Plasmodesmata can be formed in two different ways. There are Plasmodesmata that are formed during cell division when the cell plate encapsulates pieces of the endoplasmic reticulum during cytokinesis and cell division. And those are the primary Plasmodesmata. But there does exist a secondary mechanism for inserting pores into existing cell walls in the absence of cell division. And we don't know how that happens. That's really an open-ended question, like an open question in the field. How do you, like I mentioned briefly earlier, how do you poke a hole in the wall without rupturing the cell? So it's something that we're working on trying to understand. But there is a second mechanism for inserting Plasmodesmata other than those that are formed during cell division.
Okay, we're getting to the end almost of our questions, and there was another one asking about callus during PTI, pathogen PAM-triggered immunity. So is the function of PD conserved in viral and non-viral pathogen infection?
I suspect no. I suspect that the viruses so they're going to be trying to do different things So the bacteria are not going to spread through the Plasmodesmata. So they're not going to want to necessarily increase their transport. They might want to increase transport of nutrients and things like that. So there are probably mechanisms that the plants use to try and prevent that. And the bacteria themselves probably have effectors that help negate the effects of the plant to do their own thing. But I think the viruses are going to be, they're trying to do something very specific. They're trying to replicate their genome and move from one infected cell to the other to help promote their replication and their chances of being picked up by a vector and moved to another plant to spread. And so I think probably the mechanisms that are being used here are going to be different.
Yeah, but in common, I'll just add that you have the mechanisms for systemic acquired resistance that would probably be similar in those situations where you need the movement of molecules, you know, for your to induce that response in the neighboring cells. So there would be some parallels, but I think the viruses probably have a very profound, much more profound effect in that regard than would other pathogens. Yeah.
So I think we're going to take one last question because we're coming up on 12:30 on an hour and a half. So you've done a lot of stamina there, Brandon. So our last question is, the absence of virus from the apical meristem, is it connected to lack of Plasmodesmata? So that's an interesting question.
Yes.
Because we know Plasmodesmata changes developmentally.
Right. And so the meristem actually does exhibit one example where you have tight regulation of the Plasmodesmata contacts between neighboring cells. And so I think it is very well possible that it is the inability of, or the lack of Plasmodesmata that prevents the viruses from invading these instances. There are a few examples, to my knowledge, of viruses that are able to invade the meristems of plants. And I don't know what that is. I don't know if it's potentially that they're able to increase the Plasmodesmata at these specific sites, if they change something else that allows this to happen. But I do think that the inability or the lack of Plasmodesmata at the meristem is one of the driving factors for the lack of virus invasion into these tissues.
Yeah, you have these regions where, you know, transcription factors are moving through Plasmodesmata, but it's such a small restricted region so that the meristem doesn't go out of control, that
Right,
these
so
little tiny domains.
Yeah, very tightly regulated there.
Yeah. Well, I'd like to thank everybody for their questions. I'm sorry we can't get to any more. We have lots of comments in both in the chat and the Q&A. Thanking you for the very nice and very interesting talk, Brandon. And I really appreciate both you and Tessa Birch-Smith of handling all those questions. That was probably 45 minutes of questions. So, so thank you. And I really like that because that gives us a chance to interact with our readers outside the pages of the journal. So, and outside the regular publication cycle. So instead of just interacting with you once a month when our new papers come out, this gives us a chance to really engage with listeners and audience across the world. I hope that people who are interested in learning more about viruses and more about Plasmodesmata in particular will take a look at the very nice review that Brandon and Tessa wrote, and also look around a bit more in the focus issue, more interesting papers on reviews and research articles on plant virus interactions, cell biology. And in fact, we had, for a couple of issues after that, we had a few more focus issue papers coming out that were connected to that. So a lot of really good plant and interesting plant virus work coming out at the beginning of this year. So I'm going to stop there. I believe at this point, I forgot to pull up the seminar series. Here we go. I believe we do not have anything. It's August. And so 2 weeks from now, we are having a little break. We have no seminar. Everybody can go to have a little break before, in the Northern Hemisphere, we come back to classes and school things, and the Southern Hemisphere, I know you're moving on as it's winter. Anyway, so no seminar in no seminar 2 weeks from now. We'll be coming back on September 1st with a very interesting talk by Antonio Molino, who's going to be following up this, looking at the relationship of cell walls and cell wall synthesis and cell wall breakdown products in signaling between plants and microbes. So that will be, I think, a nice connection off of this. So thanks everybody for joining. I think that in the chat, I believe that Ashley has posted the link for both Brandon's paper and also for where to register for our next seminar. And if you go to that same page, that's where you'll be able to find the recording of Brandon's talk in case you wanted to re-listen to any parts or take a closer look at some of those slides. Thanks everybody for joining. And Brandon, thank you for presenting.
Yes,
Tessa,
thank you for having me.
Thank you for co-hosting.
You're welcome.
Bye everyone.